Hello students, welcome back to the chapter Sexual Reproduction in Flowering Plants. Students, in the previous video we discussed important concept by name double fertilization and triple fusion and also its significance. Whereas today in this video we are going to discuss about development of embryo and post fertilization changes. So let's begin with development of embryo. So process of formation of embryo, the development of embryo from the diploid zygote is called as embryogamy. As we have understood in the previous videos, this diagram is of LS of ovule, which is having the embryo sac inside, and after the fertilization you can see the antipodal cells which are there, they are degenerating, and even the synergids will degenerate. Okay, the primary endosperm nucleus will also undergo division and results in the formation of endosperm. So this is a first male gamete which will fuse with the egg, making syngamy possible. See there will be formation of zygote. Okay, whereas the second male gamete, okay, will fuse with the primary secondary nucleus and results in the formation of primary endosperm nucleus. Okay, now this zygote which is there, which is become diploid, will develop into embryo. So that process of formation of embryo is called as embryogamy or embryogenesis. The cells at the chalazal end which are there, okay, they undergo degeneration, which are antipodal cells, and the apical terminal cells which are there, no, okay, they will start dividing. So once there is a formation of the zygote is going to develop a cellulosic cell wall around itself and becomes a oosphere. So zygote enlarges in size and becomes oosphere. That oosphere divides by transverse. So if we consider this as a oosphere, like this it divides, okay, that is transversely, and then the basal cell, okay, will be converted into suspensor cell, whereas the apical cell will be converted into what? Terminal cell, which is also called embryo cell. Okay, so what happened, students? Zygote became oosphere, divided transversely, forming two cells. The cell towards, what is that? Towards micropyle becomes apical cell, away from the micropyle becomes a suspensor or basal cell. Okay, now what happens? So zygote which was there, that after the fertilization is termed as oosphere, where it has enlarged in its size. It can also be called oospore. Now this divides transversely. See there. Okay, but the division is not equal, it is unequal, correct? So there are two cells formed. The basal cell is the largest and it is called suspensor cell, whereas the terminal cell, the apical cell, is the smallest. Okay, and that will be called as a what? Embryo cell. So large basal cell, which is called suspensor cell, will be formed towards micropylar end, whereas a small apical cell is called embryonal cell, will be formed towards what? Interior region of the embryo sac. Okay, now further, this embryo undergoes repeated mitosis division. Okay, now the two-celled stage which is present, that is called as a proembryo. So for 1 mark you may get a question that what is proembryo? Proembryo is a two-celled stage embryo which is having an embryonal cell which is smaller in size and a suspensor cell which is larger in size. Okay, now what happens? Okay, further there will be division. So again the division takes place. Which cell will divide? Suspensor cell which is there, that is going to divide transversely like this. Okay, so here you can see, students, there is a formation of how many? 4 cells. Can you see here? 1, 2, 3, 4. And again there will be mitosis resulting in the formation of an elongated structure. Okay, so here approximately 1, 2, 3, 4, 5, 6, 7 to 8 celled structure will be formed, and those cells are going to form what? Suspensor. Okay, suspensor cells divide transversely a few times to produce a filamentous suspensor of 6 to 10 cells. Okay, now these suspensor cells will push the developing embryo into the endosperm. Okay, now the first cell which is there now in the suspensor, that first cell of the suspensor towards micropyle end, see here, this cell becomes swollen and functions as a hostorium. So this cell is larger compared to these cells, isn't it? So this cell will swell again and will start functioning as what? Hostorium. Okay, so suspensor pushes the developing embryo deeper in the endosperm for proper nutrition. So now what happens? Now the embryonal cell, okay, this is about suspensor cell. Now this is circular cell which is there, that is called embryonal cell, will divide, okay, and it undergoes one transverse and two vertical divisions. See there, one transverse and two vertical divisions, and results in the formation of how many? 4 celled stage, then 8 celled stage. Okay, like that the division continues. Okay, so embryo, now it is having how many cells? 1, 2, 3, 4. Then 1, 2, 3, 4. How many cells are there? 8 celled stage embryo it is called. Now thus produced 8 cells divide again periclinally. So periclinal like this. Okay, so again these 8 cells which are there, they are going to divide. Prior to that, 8 celled stage is called as what? Octant stage. Okay, so 8 celled stage it is, and those 8 cells are going to divide periclinally to produce proembryo. So that we are going to understand. Okay, now this is a suspensor, students, and this is hostorium, and this is the octant stage of embryo. Correct. Now 8 cells of octant proembryo undergo many mitotic divisions. See there, and they are going to produce a globular embryo first, and then the heart-shaped embryo, and finally a mature embryo. So can you see there, the cells are dividing many a times and result in the 3 stages of the embryo, and they are globular embryo, and then heart-shaped embryo, and then what? The mature embryo. So this one where the cells are just clubbed together is called globular embryo. Okay, now globular embryo to the next stage will be formed. Okay, so what happens? Now in this globular embryo, cells undergo again a division from the globular embryo stage. Again the cell division takes place. Now see, more or less it is what shape? Heart shaped embryo. So that is the second stage. It becomes heart shaped embryo, and this elongated structure is called as what? Suspensor, and the first cell of the suspensor is called as what? Hostorium. Correct. So heart shaped embryo is produced. Now from the heart shaped embryo what will happen? Okay, so again there will be cell division. So you can see the division is very rapid, and the cells also get organized. Thus the divided cells get organized to form a perfect embryo, which is called horseshoe shaped embryo. Clear? So this is embryonal axis. Okay, so this is the embryonal axis where the embryo is lying. Okay, now from the below region of the embryonal axis what will develop? Radicle, which is going to form root system. From above region of embryonal axis what will develop? Plumule, and that is going to form what? Shoot system. Clear? Now these elongated thread like structures which are there, no, these are called as cotyledons. What are cotyledons, students? First leaf of the plant. So how many leaves are there? 2. So it is called dicotyledonous, or 2 cotyledons will be there. If there is only one cotyledon, then it is called as a monocotyledonous or single cotyledon plants. Now this is called horseshoe shaped embryo. Okay, now let us see anatropous ovule showing simultaneous development of embryo and endosperm. So we understood how there is a formation of embryo and endosperm. This is a brief idea. So this is an embryo which is in heart shape, and this is what suspens are, correct? Now the endosperm is this fluid is called as endosperm. Clear? Now this region where the integuments are not covering the embryo sac is called micropyle. Okay. Now what all happened during the embryogenesis? Let's try to see. Egg was there. That after fusion was called zygote. Now the zygote divided to form two-celled stage, then octant stage, then globular stage, then triangular stage, then heart-shaped stage, and then torpedo stage, and then horseshoe shaped, that is mature embryo. Okay, so the process of the formation of this embryo is called embryogamy. Okay, now let us try to understand about post fertilization changes. After fertilization, that is the fusion of the male gamete with the female gamete, what all changes takes place in the flowering plants? First, there is a development of an endosperm. See here students, this is egg, this is first male gamete, this is secondary nucleus, and this is second male gamete, and these three are antipodal. So what happens? Second male gamete, okay, will fuse with what? Secondary nucleus. See there, what happened? There is a formation of primary endosperm nucleus, which we abbreviated as what? PEN, that is a triploid in nature. Now this is called triple fusion, or it is called second fertilization. Now this primary endosperm will develop into, okay, primary endosperm nucleus develops into endosperm. Endosperm is a nutritive tissue for the nourishment of developing embryo. So that is one of the post fertilization product, which will be triploid tissue. So this you have to remember, as you may get in the competitive exam chromosome number related questions on endosperm. You have to remember that it is a triploid, because they may ask triploid number of chromosome by giving haploid or diploid number of chromosomes. Okay, now two types of seeds will be produced, and they are the endosperm having seeds and endosperm not having seeds. Okay, so the seeds, if wherein if endosperm is totally consumed during embryo development, so the embryo has consumed the total endosperm, then such type of seeds are called as non-endospermic or exalbuminous seeds. Okay, so whereas non-endosperm or exalbuminous seeds, examples are peas, grams, beans. Go back to the topic morphology of the flowering plants. There you will come to know monocot plants, bean, pea and gram are said to be non-endospermic. Okay, so here are the examples for the non-endospermic monocot seed. Okay, now this is groundnut, which is non-endospermic. When it comes to dicot plant students, you have to remember in this way: only the castor is with endosperm, rest all are non-endospermic. Endosperm persists in the mature seed. Endosperm continues to support the growth of embryo during seed germination, and such seeds are called endospermic or albuminous seeds. Okay, so try to remember where in the seed endosperm is present, that is called endospermic seed. If the endosperm is totally used by the developing embryo, then the seed is called non-endospermic seed. So now let us try to take an example of endospermous or albuminous seed of dicot only castor. Okay, and sunflower and coconut are the Examples for endospermic seed. Maize is endospermic, it belongs to monocot category. Okay, then wheat endosperm. So in this video we discussed about the development of embryo and post fertilization events, whereas in the next video we will discuss about types of endosperm. Hope you understood the contents of the video, students. Thank you.